Our next speaker is Zib Bar-Joseph from Carnegie Mellon University. And he'll be speaking about his project, which is titled Effective Cocktail Treatments for SARS-CoV-2 based on modeling lung single cell response data.
Great, thank you. So we just discussed, I think, the issue of vaccines and our hope that vaccines will basically roll out and will impact the spread of the virus. But even if vaccines are successfully rolled out and fast, we know that even the best vaccine data suggests that it's only 95% effective. And so there will still be a pretty significant portion of the population. Now, we hope maybe to have herd immunity, but still there might, we still might have a COVID with us for the foreseeable future. Again, not at the levels that it's now, but still as a disease. So another approach to treating the disease is to develop drugs that would work for people who are actually infected, whether they were vaccinated or not. And this is the goal of our project. Our project looks at molecular data and tries to model the way cells respond to the SARS-CoV-2 virus in order to identify, first of all, the key pathways and proteins that are activated as part of that response. And the next step, maybe try to find some treatments. And the cocktail idea is that we are not sure that targeting individual proteins would be the best approach. Maybe we need to target multiple proteins. This is a joint effort. So in terms of the overall project, it's joint with Darl Kutten, who is a physician, a lung physician at BU, and he's the one that is doing the experiments. And a group at MIT, Regina Barzila and Tommy Yakula, who are working on predicting treatments for specific proteins, and a set of postdocs in my group that are working on the modeling part and identifying the pathways and genes that are activated. So Jose, June, and yeah. So the overall problem that we are interested in is slightly different than a lot of the focus on the molecular. So a lot of the focus has been on the two proteins, or maybe now a few more, that are responsible for the virus entry into the cells. And we know that most of the vaccines, definitely the RNA vaccines, are targeting those proteins, especially the spike protein. But the virus has many other proteins, on the order of 30 proteins, and most of them are not used to enter the cell. They are used once the virus is inside the cell and they interact with other components of the cell. Of course, the virus cannot replicate itself. It needs to use the cell components. It activates innate immune response and later an adaptive immune response. And so the question that we asked is whether we can focus on the activity that the virus is activate, or what the virus is doing inside the cell, and try to understand exactly what are the proteins that the virus is activating in the host, in this case in humans, specifically in lungs, and what are the pathways that are activated. And then the question is, if we can find them, is there any way to intervene and stop the virus from using the pathways that it needs? Or an alternative is extend or improve the ability of the innate immune system inside cells to fight the virus. Now, if you look at any pathogen that enters the cell, right? So you have a pathogen that connects to the cell membrane. Again, we know that with this, it has the spike protein or some other proteins, but basically it enters the cell. And once it enters the cell, right, it can replicate itself. It generates a set of proteins and expression values, and expression basically expresses some of the genes that are encoded, and then they are translated into proteins. And a number of networks and pathways are activated in order to get to that level, right? So once it can replicate itself, it goes out, infects other cells, and the process continues. In the case of SARS, it's also killing some of the lung cells. But generally speaking, in order to be infectious, in order to get to the numbers that we see in patients, it really needs to use the cell machinery and make sure that the cell doesn't fight it, or the immune system is not enough to control it. So what we are interested in is understanding what's going on inside this interaction ball, which is basically the set of proteins from the virus that interact with the set of human proteins, and how this affects the transcription or the activity of other proteins in the cell and in the virus. And for that, we actually do have some new data sets that have been produced over the last several months now, including a large effort from UCSF and from other places, where they basically try to profile or identify all the human proteins that interact with the various virus proteins. So in this case, around 20 or 30 proteins, viral proteins. And for each one, we have a set of human proteins that have been identified. This is usually in vitro experiments, so not inside humans, but in the outside of them. But still, it's a good approach, or it's a good approximation, sorry, of the a set of proteins that interact directly with the virus. So this is one source of data that we can have about the activity of the virus inside the cell. Another source of information, which is even more more widely available, is gene expression data. Most of the gene expression data we have right now is gene expression data from individuals who have been infected with the virus, and then they draw blood from them and they can profile them. The challenge with that data, of course, is that it's usually you know, these individuals had to be infected and then they had to be identified as infected. So it takes a few days from the time they are actually infected until we actually start collecting the data. So it's very hard to model the initial entry of the viruses into the cells using this data, but at least this data is available. And that's one of the challenges we wanted to deal with. How can we generate data that actually does capture the very early events that the virus activates inside the cell? Okay, so this is, even though some data available, not all the data that we care about. Okay, so once we have this data, what do we want to do with it? What we want to do with it is to reconstruct or to identify the networks that are activated by the virus. How do we do that? So we developed over the last several years a number of different methods that take time series data. So this is data that is sampled over time, including data from single cells, so gene expression levels or the levels of genes inside cells, either bulk, which is basically measuring some averages of cells or individual cells, and then coming up with models that explain, first of all, how these cells or individual cells are affected, and identify also the pathways and the regulators that are in charge or that are activating the responses that we observe. So this is called a regulatory network. This is a network of that leads to the activation or the activation or repression of genes, basically the level, changing the level of genes. And we have ways to take time series data and incorporate other types of information about humans. So this is inside the human, that we can actually identify the specific genes that are activated, some way to order the cells and the networks that they activate. And this is done, again, this is a machine learning project. So this is done with a model called CSHMM, Continuous State HMMs. And the idea is that we want to use HMMs because they are very popular and successful for time series data, but unfortunately HMMs usually have only a discrete number of states, whereas in this case, we believe that the response is continuous. So the question is, how do we extend HMMs to be able to accommodate continuous values? And it turns out there are ways to do it. Of course, we have to make some assumptions and some restrictions, but we can do that. And using this model, basically, we can take a set of individual cells and identify trajectories for the cells and for the genes without restricting them to a relatively small number of discrete states. So this is for the regulatory network. Another part of the response is something called the signaling network. So regulatory deals with the activation of genes, turning a gene on and off, but there is before that usually, or after, depending on the exact protein, there is something called the signaling network in which the levels of these genes do not change, but their activity can change. So a protein, even if it's present, can be off, not doing anything until it's turned on by another protein. Or a protein cannot do anything until it interacts with another protein, they form some dimer, and then they can work together. So this is called signaling network, and that's another activity that the cell needs to take in order to either respond to the virus or be used by the virus to replicate the virus and generate more. So this is another network, and we have some other ways of modeling this network. Basically, it starts with proteins from the virus itself. As we said, they interact with proteins in the host, and these proteins in the host go and activate some of the genes. And so we have some computational models that try to identify the most likely paths that go from the virus proteins to the host proteins and later to the regulatory network in order to identify what causes the gene expression response that we see. So this is the computational models that we have been developing, and some of them we had to adapt for SARS because, for example, now we can also use information on the virus expression rather than just looking at the host expression. Because, for example, if we have now a RNA-seq, before we initially, I mean, a long time ago, we had microarrays, now we have RNA sequencing, and now we have single-cell RNA sequencing. The advantage of that is that we can actually find, in addition to the host genes, we can also look at the virus genes and build a model that combines the levels of the virus genes and the host genes together. And then we put it all in a joint framework, which iterates between finding the regulatory network, finding the upstream signaling networks. They have to agree because usually the endpoints of the signaling network are the starting point of the regulatory networks. And so after a few iterations, the model converges and we get a model that tells us what happens from the viral, how the signal propagates from the viral protein to the observed response that we see. So this was the goal of this project. And of course, computationally, we wanted to extend some of the methods, but primarily apply them to SARS-CoV-2. And then once we identify, we also wanted to come up with ways to identify drugs for this. As I said, one of the major challenges we had is how do we get data about the initial infection of the cells? And it turns out that we just finished optimizing a protocol that takes skin cells, turns them into stem cells. This is called induced stem cells. And then it can turn these cells into lung cells. So now we have a way to take cells from any individual, it doesn't matter, and turn them into lung cells. And then we can infect these lung cells in the lab and see what happens to them very early. So basically right after infection, a few hours after infection and so forth. So this is what our collaborator Darrell did, and they basically generated, and they just published a paper in which they generated a response expression data from these iPSCs or lung-derived cells. And you can see, I mean, you can do initial analysis on this. This is bulk gene expression data. We're still, we have single cell from others, but not from this. This is bulk. But still, you can see that a lot of the genes that come up are known immune response genes. Again, I won't go into the details, but there's clearly a difference between the genes that you see responding after 4 days compared to non-infected cells. And even after the first day, you can see already some response. So this is the data that we have. And using this data, and as I said, some information about which protein interact with the virus and a lot of other general information sources about protein interaction and protein DNA, which are not related to SARS, but are generally useful for constructing network, we were able to construct the following network. So what I show here is our reconstructed SARS response network. And all the red proteins are proteins, human proteins, that directly interact with the virus. So these are various host proteins that directly interact and we believe are playing a major role in the response or the activation. The green proteins are what we call intermediate proteins. These are proteins that don't directly interact with the virus, but are used by the red proteins in order to get to the transcription factors. These are the ones that activate the genes. So these two sets are not directly interacting with the virus, but indirectly are responsible for the response, where the blue ones are regulatory transcription factors that regulate some of the response. And this is, so this is the signaling network. We also have the regulatory network that is associated with this. And these are some of the transcription factors that we identify. And these are the responses that we see for the data that we analyzed. You can see some genes are going up, some genes, this is genes, these are transcription factors. Some genes are going up, some genes are going down. And you know, this changes, of course, for day 1 and day 4. Okay, so this is the model that we had. So we had a set of proteins. We had a set of transcription factors, and the question is which. I mean, but this is on the order of 100 proteins. Which one should we focus on? Which are the most important ones for us to try and test basically specific drugs? So for this, we did a couple of things. First of all, we did some network analysis. I won't go into the details, but we have methods of analyzing these graphs to identify bottlenecks, basically proteins that a lot of the information has to flow through them, which we believe are important. The nice thing about these methods is that it's very easy to make them combinatorial. So we can look at the top 2 proteins or the top 3 proteins, and this is where cocktails come in, because we can target not the top protein, but the top protein pair, the top triplet, and so forth. Another thing we looked at is underlying conditions. So we know, and it was discussed, that comorbidities have a big impact on the ability of SARS or of the virus to basically impact the individual. And we can look at the expression response to these, or the expression levels following in lungs, following these conditions, and identify proteins that are activated by the comorbidity conditions and are also key in our networks, and take the intersection in order to identify higher priority. And we have done this, and we see pretty good overlap between at least some of the comorbidity proteins and the proteins in our network. And we can do an analysis of the genes, and you can see that a lot of the genes we identify in the network are indeed known to be related to viral processes. Of course, they were not really known to be specifically related to COVID, but it makes sense that they come up in the network. So the next step for us is we divided it into 2. First, we wanted to test some of these in the lab. Basically, what we do is we take the stem cells that I mentioned, these iPSC-derived cells, infect them, and then treat them with the drugs that we predict, and see if this affects the viral load. So we can check how many viruses are produced after some time, and then test whether the drug really works to reduce viral load. And we selected a few proteins based on the intersection of all of these, plus some other information. And some of these proteins have approved drugs. So not all of them. A lot of them don't have approved drugs, and I'll talk about it in a second. But some of these proteins do have known drugs. And we are now, again, unfortunately, I don't have results for this. But we are now, Darrell is now basically testing some of these, because we already have the drugs, on the stem cells. And of course, I mean, I won't go into the deal, but it's not trivial to do these experiments. You need a very secure lab and so forth. But the other approach we took is, okay, so if we have drugs, obviously we are going to use it. But the other approach we took is, what about proteins that we think are very important, but there's no drugs available? And this is the second part of our project. And for that, we are developing methods based on message passing neural networks, which are trying to predict the impact of certain molecules or drugs that already exist but are not known to, or we don't know all their targets, on new targets. So if we have a set of identified proteins and we have a set of molecules that we don't know, or we are not sure what are their targets, we want to build networks that can take that information, of course, learn from known molecules and their targets, but so they are trained on that, but then be applied to new molecules for which we know the structure, but we know all the targets, in order to predict their targets. So this is the project, and this can be done for one, of course, prediction, but this can be also done for multiple. So if we have multiple proteins, we can try which of the molecules can impact multiple, or either 2, or which pair of molecules can impact best without side effects the 2 proteins that we are interested in. So this is something that they have been developing. Of course, the challenge is that if we know the targets, that's good, but if we don't know the targets, how can we predict just based on the structure? And one way to solve this problem is to incorporate, in addition to the structure, which is the input to the neural network, also information about protein interaction. And then assume that if you have proteins that interact, drugs that work well on some of them can be learned by looking at the pathways that are downstream of a specific known target of that drug. And again, this is ongoing work, and hopefully the next, if there is when we have the next the next workshop, we will, or they will, the MIT team will provide more information. Okay, so I'm out of time. So basically, what we are focused on, as I said, is trying to find cells or responses within cells and networks that are activated by the virus. In addition to the molecular data that we collect, we intersect the networks that we identify with a lot of other external information, including comorbidity. People have done some knockout experiments and knockdowns and so forth, and we use all of that. Once we identify the top genes, we are trying to do experiments to test whether knocking them or treating them is indeed going to lead to reduced viral loads. And of course, for those that we don't know what drugs we should use, we are trying to predict new molecules and then test them as well in order to see what is the best way to treat the infection. And with that, I will stop and happy to take